Now we know.
I'm going to speak about functional sites induce long-range evolutionary constraints in enzymes. But before I do that, I want to make two announcements. The first one, this works. If you like this city, we have a job posting, right, for an assistant professor in our Department of Integrative Biology. We have a very open search this year, so if your research fits into this conference, probably you can apply for the job. And then there's another announcement, which is if you like the city, come back next year for SMBE 2017, which I'm organizing, and it will be just across the street. It will be in the Marriott Hotel, so you can sleep in the same building as you have the conference presentations, which I personally care for a lot, because it's fun. Okay, so those were my quick announcements, right? This is actually right after evolution content, so you can do both. Okay, so I'm going to ask this particular question. How does protein function constrain site-specific evolution? That's sort of my broad picture question I'm currently after in the lab. And what that means in practice is we want to explain this. Right? Let me see if I can get rid of the pointer. Okay, so this is an alignment of many different homologous proteins. And if you look at this, it just looks like a mess, right? But if you stare at it for a while, you notice, well, there's some columns that are completely conserved, right? They don't change at all over deep evolutionary time. So this is from the entire tree of life, any homologous sequences that we found. So this goes way back in Joanna's evolutionary time graph, all the way to the left, right? And then other columns are mostly conserved, right? You can see this in the graph by letters changed, but the color is mostly the same, right? This is mostly red, this is mostly green with some red, and so on. And then there's other columns that are totally variable, right? And again, you can see that quickly by just looking at the colors, and they're all over the place. Now the mystery, maybe, if you just look at this, is, well, there's really, you have columns that are completely conserved right next to columns that are quite variable, right? And that's really the challenge. How can we explain that there's a column of, say, all D's right here, and then just 2 or 3 steps over, it varies quite a bit. Well, it becomes much more understandable if we look at it in the context of the structure. So now I'm showing you the variability at sites mapped onto the structure. So that is the structure. So that the protein is actually an E. coli exonuclease. And what I'm showing you here is the evolutionary rate. So that's a measure for how variable that site is. So white is very variable, high rate, and brown is low variability, low rate. And what you see is on the surface, you generally have a high variability. So this is a surface residue that I have highlighted here. And then in the core, you generally have more conserved sites. And the interesting thing is, if you, for example, look at this helix, you go back and forth, right? Variable, conserved, variable, because you alternate between exposed and varied, right? And so in the alignment, it makes no sense. But in the structure, it makes perfect sense, right? So that's something that people have known for quite a while, that varied residues are more conserved than exposed residues. But then, of course, we also have protein function, right? So that is indicated here by these catalytic residues, which are shown in white, not because they're highly variable, but just because they're what? Notice that this is kind of an off-white, and this is a real one. Right? So we have catalytic residues which we know are highly conserved, right? And so now you realize there's this confounding thing. Well, is this side here brown because it's in the interior of the structure, or is it brown because it's really close to a catalytic residue, right? And so that's the specific question that we looked at in this particular study. To what extent do catalytic residues constrain the evolution of sites in a protein? And so to do that, we need to somehow measure where we are relative to a catalytic residue, and we simply measure the distance, right? So from each side in the structure, we measure the distance to the nearest catalytic residue. So it's not necessarily the same, right? Say from this one, we're over here a little closer, and from this one, we're over here a little closer. So that gives us this distance metric, and then we look at the conservation of sites as a function of this distance. And we do this in over 500 protein enzymes. And the first thing you can do is you plot evolutionary rate as a function of distance, right? And so then you get this result here. Down here we have the distance to the catalytic residue in angstroms, and here we have the relative rate. So 1 means it's the average rate of the protein that you're looking at, right? 0 means it's basically conserved, and then above 1 means it's faster than average. And you see this almost linear dependence of the distance to the nearest catalytic residue out to about 30 angstroms, and then it levels off. The leveling off, though, is a bit of an artifact, because if you look at how many residues you have captured here, you have captured almost 80% of the average structure by this point here. So this is the number of residues that you find as a function of the distance to the catalytic residue. And by about 30 angstroms, you've captured 80%, right? And so this tail here is just really thin. There's not a lot of data, and that just causes this artifact. And then what you're seeing here, these dashed lines, that's an important concept that I'll talk about in a minute. Those are the shells. So you can think of 0 means you're a catalytic residue, right? And then there's nearest neighbors, right? That's the first shell. So this dashed line here is the boundary between a catalytic residue and its nearest neighbor, right? That's why we don't find anything here. That's about 7.5 angstroms. It's a distance from one amino acid to the next amino acid, right? So catalytic residues, nearest neighbors, second nearest neighbors, third nearest neighbors, fourth nearest neighbors, fifth nearest neighbors, right? So you can go out 5 neighbors and you still see an effect from the presence of the catalytic residue, right? That's why we call it long-range conservation gradients. Okay, but now we have this problem that we are confounded by whether you're in the interior of the protein or on the surface, right? And it turns out catalytic residues often are in the interior of the protein, so we don't necessarily know whether this is a true effect from the catalytic residue, or it's just because we happen to start in the interior and move up. So to address that question, we built combined regression models. So what I'm showing you here first is the average empirical rate, right? So this is the rate that you measure as a distance from the nearest catalytic residue. Now not in angstrom, but in shells, right? So number of nearest neighbors, first, second, third, and so on, nearest neighbor. And again, this is just this linear increase. The further you go away, the higher your rate. Now we can make a regression model where we try to predict this empirical rate, but use only the structural variables, use only whether you're in the surface or on the core. And this looks something like this. So WCN and RSA are 2 measures for how buried a residue is. RSA stands for relative solvent accessibility. Are you on the surface or on the core? WCN, weighted contact number, measures how many amino acids are around a given amino acid, so that also measures whether you're in the core or on the surface. And you see that model indeed does give you an increase with distance, but it's really quite off right at the catalytic residues and at the nearest, second, third nearest neighbors. And then further out, it gets it actually wrong in the other way. And this is the mean of the prediction, right? So we're not asking whether every single site our prediction is correct, but on average, is our prediction correct or not? And on average, it is not correct here and it's not correct here. Then if we throw distance in, we get almost exactly correct on average, right? So this distance here, this distance here, this distance here, that's the difference between having the distance to the active site in the model versus not, right? So there's a clear effect in addition to being buried or exposed that comes just from the active site. And an additional way to look at it is to say, okay, we can specifically look at structures where actually the active site is on the surface, and then we know for sure that there isn't this confounding effect. And I'm just showing you two examples here. So these are the individual correlation coefficients. K is the rate, so the correlation between K and distance is quite high here, 0.6 in this particular example. Correlations between distance and WCN or distance and RSA are quite low, because distance, active site is on the surface, so distance to the active site does not correlate with weighted contact number or RSA. The relative rate, on average, increases almost linearly with the distance to the catalytic residue, and this is what it looks in the structure. Blue means conserved, yellow means variable in this particular case. And if we zoom in, we see very nicely how all the sites right around the active site, so this is a catalytic residue, are conserved, and the further you go away, the more variable they are, right? Second example, again, there's almost distance and rate are fairly highly correlated. Now in this particular case, rate also correlates with WCN and RSA, but again, distance does not correlate with WCN or with RSA. Rate increases linearly with distance, and this is a particularly nice example because this is a dimer, and you have the most conserved parts out here where you have the active site, and you have the most variable parts here where actually the two proteins need to stick together, right? That was somewhat a bit of a surprising result, because this means that this protein-protein binding interface actually is quite variable. And it turns out that protein-protein binding interfaces tend to be not as conserved as we may think. So that's the last slide with results that I have. If we just look at the relative rate at catalytic residues, at interface residues, and at all residues, we find catalytic residues are extremely conserved, right? Remember, 1 is the average. All residues, on average, have rate 1. Catalytic residues almost have rate 0, right? They really do not change. Interface residues, they're a little bit more conserved than your average residue, but not very much, right? Interface residues are surprisingly conserved, surprisingly variable, and we tested that in a non-enzyme data set, and we found the same result. Maybe the distance is a little bit bigger here, but it's still the case that interface residues are quite variable. Okay, so the take-home message here is catalytic residues generate long-range selection gradients covering most of the typical enzyme structure. And I don't have it in words there, but protein-protein binding interfaces are surprisingly better. Okay, so this is work that was primarily done by my graduate student Ben Jack in collaboration with Wilke in Argentina and a former graduate student, Austin Meyer. Thanks a lot, and we have time for one
So you looked at the interfaces compared to all of the residues for non-enzyme proteins, but did you also, for the non-enzyme proteins, look at just the buried versus surface variability?
So the question was whether in the non-enzyme proteins we looked at the variability between the buried and the exposed residues. I don't remember in this particular data set, but in general, every time you do that, you see a strong correlation. Exposed sites are variable and buried sites are...
So it's not just that catalytic sites tend to be buried.
So I guess the question, a different way of formulating the question would be, is it that in the enzyme data set, buried sites seem more conserved because they're near to catalytic residues? There may be a bit of an effect of that in that data set, but I think both effects are independent. Okay, why don't you set up, and if there's one more question back there.
Have you looked at enzymes where the catalytic site is changed, and to ask whether is it like maintaining the catalytic pocket, or is it maintaining specific catalytic residue?
So the question was, have we looked at enzymes where the catalytic site has changed? And the quick answer is no. Generally in this analysis we only have one structure. So in that one structure we have specific catalytic residues, and then we make an alignment of homologous sequences. But certainly if one has a case with closely related enzymes where there's different locations of the catalytic residue, so one could do that kind of analysis, but we haven't done that yet. Thanks.